Jai Hind everyone, I am Pankaj Kumar and today's lecture is about the Pentose Phosphate pathway and or it is also known as the HMP pathway, means hexose monophosphate pathway, or it is also called as a phosphogluconate pathway. Listen it properly, phosphogluconate pathway. So it is very important because the glucose 6 phosphate which is formed that have a two feet, either that can be go for the glycolysis or it can enter for the pentose phosphate pathway. What are the deciding factors when it will enter into the glycolysis, when it will enter into the pentose phosphate pathway, that we'll read in this lecture. And in this lecture, if you'll continue, you'll find why the Pythagoras, correct, you're listening correct, Pythagoras, who was a mathematician, he not used to eat the fava beans. And if you'll see the Mediterranean peoples or the Middle East people, African peoples, they are usually they are resistive to the malaria. How it can be happen and what is the relation with this pentose phosphate pathway and the malarial resistant African peoples. In this only we'll see the thiamine, vitamin B1, how it is important for the curing the beriberi and the other disease which is called as the Wernicke-Korsakoff syndrome. So let's continue with this lecture. So pentose phosphate pathway have a two fit. Either it can go for the glycolysis or it can go for the pentose phosphate pathway or the uh, it will go for the HMP pathway. So where it will be occur in our case? So it will occur in the bone marrow or intestinal mucosa or tumor cell. So what is help? How it will help the tumor cell, the pentose phosphate pathway? So important thing is that in tumor cell during pentose phosphate pathway it will form ribose 5 phosphate, and this ribose 5 phosphate is very important because it will give you the RNA and DNA. Basically it is giving nucleic acid synthesis, and as well as it is as well as it produce coenzyme as ATP, coenzyme as ATP, NADPH, NADH and FADH, DH2. So it is giving, as you can see, it is giving lot of things like ribose 5 phosphate, synthesis of the nucleic acid, synthesis of the coenzymes like NADH, FADH to coenzyme A. So these are the very important things. Here you can see the pentose phosphate pathway is divided into the two parts, the oxidative and non-oxidative phase. So in oxidative phase, the glucose 6 phosphate is finally converted into the ribose 5 phosphate, which is used for the nucleotide coenzyme and DNA RNA synthesis, as you seen before. The byproduct is what? The NADPH, which is the very important, and the NADPH, how it is going to be helpful, we'll see in few minutes. So let's see, glucose 6 phosphate is uh, converted into 6 phosphogluconate by the accepting of the electron by the NADP plus. It is giving the NADPH which convert the GWS to the GSH, and like this is converting the oxidative mm, glutathione to the reduced glutathione. Then phosphogluconate is converted into the ribulose 5 phosphate. Again it is reducing the NADP plus, and ribulose 5-phosphate is converted into the glucose 6-phosphate. We'll see in the non-oxidative phase, and ribulose 5-phosphate is giving ribose 5-phosphate. So basically the concept is that the role of NADPH, we have to understand the role of NADPH. What is the role? As the radiations will come on this RBC, okay, suppose radiation is coming on RBC, it is coming on the lens, or it is coming on the cornea. So in these all cases, the RBC or the lens, these all are directly in contact of the UV rays. So this all will form, this all will form superoxide, superoxide, or it will form free radical. Now this free radical is very harmful. Okay, this free radical is very harmful for the cell. It can damage the RBC. So in this case, we need NADPH, which will act as a electron donor. It is as a electron donor, and it will minimize the risk of the damage. So finally, it will prevent DNA, RNA, etc. damage. So this is the important thing about the NADPH. So basically, it is a electron donor which will help in the reducing the risk of the damage of the tissues and all. So whenever you will find there is a NADPH and by NADP plus ratio is high, or so whenever NADPH by NADP plus is high, or GSH GSSG is high, then it will reduce this, both will prevent oxidative damage in cell. So you can see this image is taken from the Leninger, and this is used for our education purpose. So learning purposes, it is very helpful. You can refer this book as well. So let's continue with the oxidative phase. Now we'll see oxidative phase. So oxidative phase started with the glucose 6-phosphate. So glucose 6-phosphate is converted into 6-phosphogluconate delta lactone in presence of glucose 6 dehydrogenase. This is the very important enzyme. You remember this, and this conversion reduce NADP plus to NADPH. Now glucose 6-phosphoglucono delta lactone is converted into 6-phosphogluconate by the help of lactonase enzyme, in which one water will be used and one magnesium is used. So 6-phosphogluconate is converted into ribulose 5-phosphate in the presence of 6-phosphogluconate dehydrogenase. Here also one NADPH is forming, and now ribulose 5-phosphate is converted into its isomer, that is ribose 5 phosphate. So the outcome is what? The outcome is the NADPH. And you know why NADPH is very important? Because it is preventing the oxidative damage. How it is preventing the oxidative damage, we'll understand after the non-oxidative phase. Non-oxidative phase is basically conversion of the pentose phosphate to the glucose 6 phosphate. So what is the our pentose sugar? The ribulose 5 phosphate. So we have to convert ribulose 5 phosphate to the glucose 6 phosphate. There will be several steps. We'll see one by one. The first step is the conversion of the ribulose to xylose 5 phosphate. So here it is. Let's see this ribose 5 phosphate is converted by the isomerase and epimerase. Then it is forming xylose. This together it is with the trans-ketolase enzyme. It is converted into the pseudoheptulose phosphate and glycerylate 3 phosphate. Together they are forming a 6 carbon fructose phosphate and 4 carbon erythrose phosphate. This 6 fructose phosphate is directly giving the glucose 6 phosphate by the isomer, and then erythrose phosphate is again binding with the xylose phosphate with the help of trans ketolase. It is giving fructose 6 phosphate and glycerylate 3 phosphate. Remember it, the aldolase is giving the erythrose and aldehyde, the glycerylate 3 phosphate is given by the ketolase enzyme. Now glycerylate 3 phosphate can give fructose 6 phosphate as by following enzymes, and then fructose 6 phosphate can give glucose 6 phosphate. So this is the regeneration of the glucose 6 phosphate. So it is forming a one complete cycle. You can follow this, like how many carbons compound is there. So this is the ribulose 5 phosphate, this is xylose 5 phosphate, then pseudoheptulose glycerylate, then fructose erythrose, then again xylose, and then it is giving glucose 6 phosphate, and then again it is giving glucose 6 phosphate. So you remember this charts as per your convenience. That is going to be helpful. Now we'll see how this NADPH is used for the reduce preventing the DNA damage and the reducing the oxidative stress of the cell. So in front of you, one picture is there, that is Pythagorean, and this is the fava bean. So now if you'll see the fava bean, it is a vegetable which used to be cooked in so many houses, but Pythagoreans didn't like that. Why he didn't like that? He will become ill. And what is happening in this? Whenever he will take the fava bean, the H2O2 will form in this body, and either H2O2 will form or O2 radical or just superoxide will form. So these oxides have to be clear. Okay. So in the presence of NADPH, NADPH, this will be converted into the H2O plus O2. But if the NADPH is not there, and if the glucose 6 dehydrogenase enzyme, what? Glucose 6 phosphate, 6 phosphate dehydrogenase. If glucose 6 phosphate dehydrogenase is not there, then this NADPH will be not there. If this NADPH will be not there, then H2O2 will be not converted into the oxygen and water. So that will accumulated more, and then cell will go for the oxidative stress. Then in this case, what is happening whenever the toxic like H2O2 or superoxide will increase, our RBC will start lysing. Okay, so this RBC becomes sad, and finally it will lysed. Okay, so this RBC will be start lysing. Finally, this is causing a disorder which is because of the lack of the glucose 6 phosphate dehydrogenase. Because this is important for the NADPH production while converting the glucose 6 phosphate into lactones or glucose delta lactone. Okay, here 6 phosphogluconolactone, and if this enzyme is not present, then it cannot give the NADPH. This is the whole concept. So usually this person will be happy only whenever the glucose, they will not take a fava beans, because fava beans is giving this oxides, and it is forming a uh, toxic more like more oxidative uh, environment for the cell, and that is a toxic for the cell, and lysis of the cells will be occur if NADPH is not there, then only. And usually the glucose 6 phosphate dehydrogenase deficiency is asymptomatic, and the person will be not getting any symptom until unless they will not give the oxidative stress to their cell by the fava bean or by other means. So other means are what? If you will take the if you will take anti-malaria anti-malaria drugs. What are the anti-malaria drugs? You can I will give you one example. Primaquine. Okay, Primaquine. So in case of Primaquine, it is also increasing the H2O2. So why it is giving? It is giving the oxidative stress to the oxidative stress for Plasmodium. It is a protozoa which is causing a malaria. So malaria will decrease. So malaria will decrease. Okay, this is the concept. If the giving the Primaquine, then malaria will decrease. But if the person doesn't have this and doesn't have this dehydrogenase, and you are giving the anti-malarial drug, then the person will be become worse. That person will health become worse, and the person may be this disease, these drugs may be fatal to that person. So in that case, if any person without the glucose 6 phosphate dehydrogenase, then that person doesn't require any Primaquine type of drug, and they never get the malaria. They will not get the malaria disease. This is the also important point. So more than 400 millions, mostly in the Middle East, African, like malarial prone area, are affected with this disease, and the disease is called as a favism. This disease is called as a favism, in which RBC will be ruptured. So this is about the Pythagorean story, and now we'll see the thiamine story, like how vitamin B1 thiamine is useful for the curing the disease like a beriberi and all. So thiamine, which is also a vitamin B1, it forms thiamine pyrophosphate, which is a cofactor for the trans ketolase and pyruvate decarboxylase. But important thing, pyruvate decarboxylase we didn't see here. We see in trans ketolase, which is forming the glucose 6 phosphate aldehyde. Okay, here it is forming glyceraldehyde 3 phosphate, and here also it is forming glyceraldehyde 3 phosphate. So trans ketolase is requiring the vitamin B1 for its functioning. It stabilizes the 2 carbon ion in the reaction. Uh, this is the very important thing. The first and third step of the oxidative phase is pyrophos pentose phosphate pathway is irreversible. All enzymes of the pentose phosphate pathway located in the cytoplasm, and glucose 6 phosphate partition in the pentose phosphate pathway or a glycolysis. Like it is optional for the glucose 6 phosphate where it have to be go entered. Either it will enter into the glycolysis or either it will enter into the pentose phosphate pathway. So it depends totally on the NADPH concentration. If NADPH concentration is very high, then it will uh, then it will activate the glucose 6 phosphate dehydrogenase, and then it will convert the glucose 6 phosphate into the 6 phosphogluco uh, gluconolactone. Then if the NADPH is if the NADPH demand is very high, then it will, if demand means NADPH is very low. Okay, so if the NADPH demand, or I will tell you like this, suppose NADP plus is very high, then it will activate the glucose 6 phosphate dehydrogenase will, sorry for the bad handwriting here, it is activated. So it is activated. Once it will be activated, then it will influx of influx of glucose 6 phosphate. Okay, it will influx of glucose 6 phosphate will be occur. It means it will go for the pentose phosphate pathway. Got it? If the NADP plus is very low, what is mean? If this is low, it means NADPH is high. Okay, it means NADPH is high. Here NADPH will low. So if this is the case, then what will happen? Then glucose 6 phosphate dehydrogenase will inactivate. It will become inactive. Then there is a no influx of glucose 6 phosphate in the pentose phosphate pathway. So it will enter in glycolysis. Okay, it will enter in the glycolysis. This is the important thing. Okay, so this is all about the today's class. You can find the berries in symptoms. Our body fluid will be swelling and then pain, paralysis. Without treatment, the person can be good for the death. And Wernicke-Korsakoff have a voluntary movement problem. Eye movement will be not occur properly, and neurological defect will be occur. This beriberi or the Wernicke-Korsakoff, both diseases because of the deficiency of thiamine, and thiamine use is help in the transketolase enzyme. Thanks for watching. We'll meet in the next video. If you have any doubt, you can ask me, and you must solve the questions which is given in the description box related to you. related to this topic, and it definitely it will help in your examination for what you're preparing for. So thanks for watching. We'll meet in the next video. Jai Hind.